All right, so hello everyone. Welcome to week 2 of our PSPablo. Hope week 1 was good for you guys, and we'll get started. Can you guys all see that? Or yep, awesome. So this week you guys are going to be covering a couple of concepts, some really key ones that are sort of going to flow on till years on end. So you got the central dogma, which we'll talk about a bit later. You've got acid base, so like a bit about like the body's pH levels and stuff like that. You've got stuff about enzymes and cellular energetics, and you've got a lot about fuel molecules, and that's going to be a big unit that continues to week 3, I think, as well. So we'll just we'll go from there. So I'll start off with the central dogma, and what the central dogma sort of is. It's the theory that genetic information is encoded and is self-replicating in DNA, which is then transcribed to RNA and then translated to proteins. So why this is so important is because pretty much our cells do everything or like our body, everything that happens sort of is by us making proteins in the end, which are the functional sort of things that do things. Yeah, that was really well explained. So pretty much our DNA, it replicates itself and makes those copies, which then get turned into RNA, which is sort of like the messenger or the conduit between the DNA and the proteins, which then goes to the ribosome, and we get the proteins actually being made. So yeah, that's sort of like the 3-step process, and that's what we call the central dogma. So we'll start off with DNA, and there's a big concept called the replication fork. So I thought surely we chuck in a meme because it's funny that way. So DNA replication is pretty much, it's that thing that Mitch talked about last week. You know how we said there was a G1 phase, S phase, and G2 phase in mitosis? So that S phase is where 2 identical copies of DNA are made, so that when the 2 cells are made, you have DNA in both. And so that's the process of DNA replication. So you get one old strand and one new strand becoming this new formed DNA. And that's why we call it a semi-conservative process, which means it's like not fully conservative, not just the old stuff, but it's half the old stuff and then one new strand. And this all happens in the nucleus. So we'll sort of try breaking down the whole process into 7 main steps, and we'll show diagrams at the end after, so we can sort of just solidify what's going on. But we start off with an enzyme called helicase, which just comes in that 2-stranded DNA and breaks it apart, so we can start replicating the 2 ends. Then we've got these little proteins called single-stranded binding proteins, which just go into the 2 strands that are opened up now, and they keep them open. Because if you think about it, all we're doing is breaking the hydrogen bonds. So there's nothing stopping the 2 strands from going back together. So we need something that sort of holds them out so that we can do the replication. And that's what the single-strand binding proteins do. Then we have something called a primase, which is on its own like pretty important, just because our other enzymes sort of needed something to grab onto to like start the whole process. And so primase is that sort of little bit that things can sort of grab onto so they can start the replication process. And it binds at something called an area we call the 3' end. So like the whole strand itself, we say one end's 3', one end is 5'. So it just starts with the 3' end. You sort of just need to know those sort of numbers and where they fit. So we have an enzyme called DNA polymerase 3, and that encloses the strand and it starts replicating. And how it does that is it looks at the base that it's looking at. So you know how we have like 4 different bases in DNA, like adenine, guanine, cytosine, and thymine, I think I forgot. Yep, thymine. It sort of sees what's there and gets a base that's sort of like the opposite of it and attaches that onto it. And so you get a strand being formed like that. And we make it a 3 to 5' direction. So that's just why those primes are important. Step 5 is we get something called RNase H, and that just removes the primers because we don't need them anymore. We've already attached, we've already started making the strand, so we get rid of them. Then that's on like the strand that's like, there's because you know how like we have 2 strands for DNA and we break them apart. One's going 3 to 5', one's 5 to 3'. We call the 3 to 5' one whatchamacallit, we call the 3 to 5' one the leading strand because it's just the same way that DNA polymerase is going to move. But the issue is the other side is called the lagging strand, which is 5 to 3'. It's the opposite way of what DNA polymerase sort of likes to move. And so the issue with that becomes that like DNA polymerase can't just go across it like as usual. It sort of does it in little bits and blobs, and we call those bits and blobs Okazaki fragments. And so the issue with those little bits and blobs, as we said before, is they're going to need each of them a separate primer. You know how we have the primer to start everything? We need all these little bits of primers all over the strand. And so you have like sort of a fragmented sort of arrangement going on there. And so that's why we need something called DNA polymerase, which sort of removes those primers, and then another enzyme called DNA ligase, which fills in those little gaps. So eventually we do get 2 full strands, but it's just a bit more work. There are other little things that sort of aid the process. They're not as important, so I've sort of just chucked them on the side, but you've got things called sliding clamps, which aid the DNA polymerase 3 to attach from, attaching to the Okazaki fragments. And then you've got topoisomerase, which is a really big sort of protein, which just keeps the DNA helix unwound, just like those little binding proteins as well. So this is a really sort of nice diagram to show everything. That's that big molecule, that topoisomerase we talked about. Those are the little single strand binding proteins, keeping that from annealing together. That's helicase, which just comes through and rips, the helix apart, sort of thing. And those are the primers that sort of attach everything to. In your own time, you can sort of get your head around sort of this concept, I guess. Another nice diagrams here. And that sort of just also shows that whole semi-conserved nature of everything, the old strand and the new strand. Cool. So now we sort of like, it's sort of a side sort of concept, but due to the Okazaki fragments being like all like, disjointed and stuff, we get a bit of them that's sort of overlapping, and there's like some bit of the lagging strand that can't be sort of filled up, and we call that an overhang. And why those are important is because pretty much every time that lagging strand is going to be shorter and shorter and shorter. So just in general, our DNA slowly over time starts to degrade. It gets shorter and shorter and shorter. So that's why our awesome body has decided to make things called telomeres, which is just extra bits at the end of each chromosome, which we don't need. So if we get rid of them, it's sort of okay. The issue here is that things like cancer abuse the fact that we have these things, because they're pretty much like the age of the cell. The shorter your telomeres are, the more you've been replicated, and eventually the body knows to get rid of these old cells because they're problematic, so we just remove things that have short telomeres. Cancers are pretty annoying. You'll learn more about this later, but they sort of reverse make telomeres so they look younger. So they have anti-aging cream, and so like you get like sort of cancer cells that just don't die because the body doesn't think they're old. So yeah, the good thing about our body is though is we have proofreading mechanisms. So we pretty much have a way to make sure errors don't occur as much, because obviously with errors you get cancerous cells and you get problems all around. So we have 3 different sort of steps that we can proofread this replication. The first one is DNA polymerase itself, when it's making the new strand, detects issues and makes sure like to remove incorrect nucleotides. So pretty much it proofreads itself, which is really great. Another thing we have is mismatch repair, which is pretty much straight after replication, proteins cut out incorrect bases from the DNA strand, and DNA polymerase just goes back and replaces them. And the final one is excision repair, which is pretty much where it constantly happens, and we just replace any nucleotides that aren't necessarily wrong, just that are prone to mutation. And we use DNA polymerase 1 to do that. So that's step 1. It's pretty heavy, but that's step 1, just done. And now we got to step 2, which is transcription. And we touched on this last week, but we didn't really talk about it too much because it's not too high yield. But DNA comes in 2 forms, a packed form and an unpacked form. So just remember that we're going to be in the unpacked form when we do transcription. Can you guys still hear me? Yeah? Yeah, cool, awesome. So transcription is pretty much where we synthesize RNA, which is our messenger, and then we process it. And so that messenger we call literally messenger RNA. So it's called mRNA. And it starts off the process with something called initiation, which is where the DNA helix unwraps again, just like in replication. And we've got transcription factors, which are these really nice little things that we don't really need to know too much about as of yet, but they bind to the promoter region, this region of the DNA strand, and they allow the enzyme RNA polymerase to just attach on, and it can start its usual growth, just like in replication, where we sort of build that new strand. But this strand is, instead of another strand of DNA, it's mRNA now. Then we've got the elongation process, which is where we actually make it, and eventually it gets to something called the termination sequence, which is the end of the chromosome or the gene, more other, where we just pretty much cull sort of the whole process, and everything rewinds, everything goes back to place. So it's much easier to understand than replication, I think. It's also regulated because, it's great like that. So we've got all these things that unwind the DNA, make sure it's all good. We have the transcription factors that we talked about before, which pretty much influence if RNA binds here or it doesn't. So we can make sure that it's more of a selective process. You've got DNA methylation, which is pretty much adding a methyl group to cytosine bases, and that stops these factors from binding, and so we stop RNA polymerase from binding just in general. And then you've got regulatory DNA sequences, which pretty much activate or repress certain sort of processes. And then you've got, after the actual transcription, you have other modification that happens as well. You've got splicing, which is pretty much removing introns, which are these like things that we really don't, like parts of the gene that we don't need. So we just get rid of them. You've got alternative splicing, which is getting rid of the bits that we do need from the gene, some bits, which are called exons. And so we can sort of code for different things from the same gene just by getting a different combination of exons spliced out. And then finally, we add something called a poly A tail and a methyl cap, and they respectively on the 3' end and the 5' end of that mRNA strand, and they just help with stability and protection of the mRNA. Just a bit of a side note here, but these things make sure, that these things are just part of the nucleus that make sure, the RNA is sort of okay. But unlike DNA, it isn't proofread. And so that's why you can have a lot more mutations and incorrect stuff happening in RNA processing. And that's why we see viruses that depend on RNA only mutating at really rapid rates, because they just don't have this proofreading. And that's transcription. So now up to the final one, which is translation, which happens at the ribosome. So there's 4 main steps to translation, and they happen sort of at different sites of the ribosome. So we're going to look at the ribosome just in a bit. But the first step is called the charging of the amino acids. So we have these things floating around called tRNAs, and they're also RNA, which is cool. And what they do is they pretty much are the collectors. They collect amino acids that are floating around in the cell, and they bring them to the ribosome. And they have bits on them that are called anticodons, which is sort of the opposite of the mRNA code, the mRNA bits. And pretty much what they do is they read that bit and see exactly which tRNA is the correct one, which will bring the correct amino acid to the ribosome. If that makes sense, please stop me if I sort of got confused. But pretty much we have these things, enzymes called tRNA synthetases, and they activate the tRNA, allowing it to link to the amino acid and then bring it to that ribosome. So the ribosome pretty much has 3 main, we call them sites: the E site, the P site, and the A site. So the A site's the first one, and it's pretty much where that tRNA brings in the amino acid. The P site is where the tRNA sort of adds that amino acid to the chain that we're sort of building. And then finally, we've got the E site, which is pretty much where the tRNA will exit the ribosome and the next one sort of come in. It's like a conveyor belt sort of thing. And so that's sort of showing you that in that diagram. So there's 3 sort of sites on the ribosome.
Just a question, Ibrahim.
Yeah, sure.
So regarding the anticodons, is it like the anticodons have the opposite base pair to the codons on the mRNA?
Yeah, so they do. Yeah, exactly. So pretty much the anticodons, that's why they're called sort of anti, because they're so pretty much complementary. So for every A, you have a U in this instance, and for every C, you have a G. So yeah.
Is there a reason why they use RNA instead of just using DNA, like uracil?
You mean with the uracil and the thymine?
Like, is there like a kind of usage for uracil as opposed to something like thymine?
That is a good question. I don't know. Do you guys have any idea? Others? Lily, Mitch, as well?
It's one of those things that you don't really need to know. Like, you don't need to know why it happens. You just need to know that it happens. Like, that's like PhD evolutionary molecular biology stuff. We don't need to know that. So just do not worry about that at all.
Sounds good.
I searched it up just now. It just says uracil is easier to produce than thymine, like uses less energy. So there you go.
Yeah,
Okay,
great.
that makes sense.
Cool. All right, we'll keep going. So step 2 is initiation, and this is a step that uses energy. So just be aware that translation is an active process. And so you begin by, that's when you bring in sort of the tRNA down. I'm sorry for those really weird A's, they're meant to be arrows, but pretty much the tRNA carrying the methionine, which is usually the starting amino acid we start with every protein, and it brings it to the 5' end, and then you that sort of directs it to the initiation factors, which start the whole process. Elongation is next, which is where the whole sort of conveyor belt process happens and you build that long chain. And then finally, the last thing is termination. So we get to a stop codon. So that's a bit on the mRNA that just tells everything to stop. That's the end of the protein, the end of the gene. So everything dissociates and you get the full protein. And that goes on to other stuff later on, which we're not going to really talk about too much today. But if it's going out of the cell, it goes to the rough endoplasmic reticulum. And if it stays inside, you've got some processing that happens inside as well. We are going to talk about, though, post-translational events, which are things pretty much the actual strand itself sort of undergoes sort of like different modifications. They're pretty low yield, not gonna lie. So I don't think we'll run through them in the class, but they're on the slide if anyone's interested. And now we'll just sort of go general stuff about DNA versus RNA, prokaryotes versus eukaryotes, which are just sort of basic stuff. In terms of the actual replication process, that's our first of the three things in the central dogma. Eukaryotes start at multiple points in that whole DNA strand, whereas prokaryotes start with one point and they just continue across that one sort of replication area. Also with eukaryotes, you have that proofreading mechanism, which prokaryotes don't exactly have. So we have less harm. And because we start at different areas as well, you get like sort of less incorrectness. Like instead of one area, if you start from one point and you go across the entire thing, that same sort of enzyme's gonna do stuff. And so if it's messed up, it's going to mess up everything. Whereas with smaller multiple origins of replication, you get less of an impact on the general sort of DNA strand. And it's much faster as well because you have different areas. And more about RNA just in general versus DNA, the differences really are that use a deoxyribose as the sugar for DNA, whereas ribose for RNA. DNA is double-stranded, RNA is single-stranded. DNA uses thymine, that's what Ali was talking about before, and uracil is the counterpart in RNA. And we just got told, which is awesome, thanks guys, that it's because it uses less energy. DNA is comparatively more stable just because of its nature and composition and all that winding and stuff like that, whereas RNA is less stable. And comparatively, DNA is much larger than RNA, which is a lot smaller. The final thing is a little bit of a side note, but it's something called Chagov's law. And he says that pretty much there has to be an equal ratio of purines, which is the A's and the G's, and the pyrimidines, which is the C's and the T's. And that makes sort of sense because, like, if you think about the double-stranded nature of the whole thing, if you're going to have one of these, you're going to have one of these. But RNA doesn't have to follow that because it's single-stranded. So that's why we call that following it or not following Chagov's law. So yeah, that's the end of Central Dogma. Hopefully that was a quick run through and you absorbed a lot of it. Even if it's just a bit, that's fine as well.
Okay, HKS and med law. So you will have these tutes. You'll have probably a HKS one every week and a med law, you'll have like 3 of those. HKS is interesting and I really loved it, but it's like, it's more important to understand the general idea of each week that you do rather than focusing on specific content. There are a few things that you do need to know. So like you need to know the Tanahashi framework, which will come up. You need to know like it will be obvious what you need to know, and we will do a thing, like we'll make it clear in our slides about like when something comes up that's important. So rather than taking down millions and millions of notes, it's more important to really engage with the sessions and try and understand the point of each session rather than just meticulously jotting down notes about what's said. So yeah, and then with med law, again, it's important to know a few things, like it's important to know consent, it's important to know what battery is, but again, when one of those few things comes up, we will do a thing about it. And we're probably going to do like a thing towards the end of the semester, which brings up, the important points for each of these subjects. So really, come with an open mind, come ready to engage to these classes, and that's probably the best you will get out of these classes. So.
Yeah, and what I was going to say as well, like what I did I would take notes like all my subjects, but for HKS, at the end of the semester, I just made like a 1-page summary of all the most important info. Like it's just not worth wasting your time going into like super, super specific details. Just enjoy My3's lectures. I don't know if you've had one yet, but they're usually pretty engaging. So I would probably just sit there and listen most of the time.
Yeah, My3's a really good speaker, so definitely enjoy his lectures. And so like this week you're doing health systems. So really, as long as you understand like the Australian health system and then maybe some differences across different parts of the world, then you'll be fine. So yeah, that's all from me.
All right, so I'm doing acid base. Hopefully, can everyone hear me? There might be a little bit of like background noise, but it should be fine. Acid base is very chill, and even though it's super important clinically, it's not necessarily super high yield. I think you might get 1 or 2 questions on it. All right. Yeah, so essentially the normal pH of the body, the top things, almost everything in bold, what you just need to know. Normal pH of the body is 7.4. So this does not mean that it's the same as like a neutral point. So like water is neutral, because water is the same as like the concentration of the OH minus equals the concentration of the H plus. And we know from like the negative pH log scale that that's 7 usually. In the body, it's 6.8, but that doesn't actually super matter. What you need to know is that at the body, your pH is 7.4. It shouldn't fluctuate beyond that kind of range. If it goes higher than that, it's going to be too basic. So alkalosis, and then acidosis is if it drops below that pH. And as we probably know from chemistry, like all those structures and levels of structure of the protons are all influenced by like the pH in which it exists in. So you want to make sure that in the body, you have a very narrow range in which it can, like this pH range can exist in which you can be like comfortably living in. So yeah, the next one, I think it's just a meme on the next page. So this is important actually to say. So I put this meme in, but just because water has a neutral pH of 6.8, that's not the pH in which our body exists in. So our body actually would, like the concentration of H plus ions and OH minus ions would be neutral, would they'd be even at 6.8, but our body exists at 7.4 pH level. Yeah, and then, okay, well, that one didn't really turn out awesomely, but that's fine. Essentially, how your blood might become more acidic or basic is just influencing the concentration of the H plus ions. So you can do this through either respiratory or metabolic processes. This stuff doesn't really matter, but essentially maybe CO2 goes in water, it kind of decomposes into H plus and bicarbonate ions. And then that would increase the H plus level and therefore decrease the pH level. Yeah, and I think that's essentially it. Oh, there's one more thing, and that's the next slide. Partial pressures, I actually don't think this will be important to you at all, but it's essentially that like a pressure in the atmosphere is made by both all of the pressures combined in a gas, and then in a liquid, that partial pressure is only in watts. So the partial pressure of a gas in a liquid is the same as the amount of the gas that would have been in equilibrium with the liquid. So it's not the same as the amount of gas that's in the liquid, but the amount of gas that's kind of in contact with the liquid, or would have been in contact with the liquid. And then last one. And then just homeostasis. This is essentially just like whatever you do, let's say I increase the H plus of your body will automatically try and decrease this H plus. So it's going to try return back to a normal state. What is this? Okay, so the liquid, so the partial pressure about the liquid, that is so the lecture we had vaguely mentioned something about partial pressure in this acid-base lecture. And it was specifically about like partial pressure of gas in a liquid. I'm fairly certain that you won't need to know this at all.
I'm not gonna lie, I don't know, like I actually cannot recall hearing that ever. So yeah, it must have been small.
Like
it's
in
very
year
small.
2, week 1, that's the first time we've heard it again since then. So like it's really not going to be coming up. Yeah,
I just added it in because in case he mentions it, but I don't actually think he will. Yeah, exactly. So don't stress about it. But it's essentially just like because of the coefficient of like Okay, yeah, I don't need to mention that, but it's essentially just based on like solubility and equilibrium coefficients, which determine how much is eventually going to diffuse into the liquid and therefore establish a partial pressure. But don't worry about it. Essentially, all you need to know is pH of the body is 7.4 and the range in which it can exist, and anything below that, acidotic and alkalotic, or however you say it, if it's above. Yeah.
Also, you probably hear us say like, don't worry about it a lot, but it's because I'm sure you already realized in week 1, there's like so much content, and you'll just come to, and at the start, you're like, I want to really understand everything and like really get into the details of it all. But what you'll realize is you just like low-key can't do that, which is why we like tell you what we think is super important, because a lot of the stuff they teach you, but they also just like don't really assess.
You also
okay.
sometimes will be,
You go, Liz.
you also sometimes will be like, how the hell am I supposed to like learn all this? But like, it's just like, you just learn, like, don't stress. Like all of a sudden it's just like in your brain.
Yeah, it takes time, I reckon. Like it like clicks later on, especially when you're like, when you get to the end of like learning. For example, if you're doing like all of this biochem, it kind of clicks a lot more when you get to the end of biochem and you're like, oh, this is how it all connects together. So that's very facts. Okay, I'm doing cellular energetics and enzymes, and a lot of this is very similar to what you would have done in bio for anyone that did bio. And also even in chem, like when you do thermochemical equations and you have like your delta, your negative delta H or your positive delta H, it kind of all follows similar principles to cellular energetics. So on the next page, these two things are probably quite important. Just, it's not hard, but they're important to understand, because when you go to look at like different reactions later on, it's useful to know whether it's like catabolic or anabolic. So basically, catabolic reactions is when we have the breakdown of macromolecules. So for example, if you have a glucose molecule and it's been broken down, that would be an example of a catabolic reaction. And the key thing to remember there is that that's going to cause the release of energy. And if you think about it, it kind of makes sense. Like if you're going from something big and you're breaking it down, it's going to be able to release. So the opposite of that, it's like cation, anion, kind of similar to that, but the opposite is anabolism. And that's when we have smaller molecules, which we call precursors, and we use them. So they would be your reactants, and we can make larger molecules. And again, if you're thinking about it, if you're going to build something up, you're probably going to need energy to do so, which is why an anabolism requires energy. So yeah, just remember those two definitions and just understand mostly like where's energy being released and where's it being used. On the green side, they don't really talk about this a lot, to be honest, but what will happen a lot naturally in like biology is that you'll see that there's this thing called energy coupling, where a lot of reactions which are exergonic or release energy will be paired with a reaction that's endergonic or coupled. So for example, you have like photosynthesis and cellular respiration. I think that's a good example. Well, if you see on the diagram, we have like the energy that is released from one reaction will be used in a like coupled reaction, because then at least no energy is going to waste. So one thing that's really important for you to remember is like ATP can be broken down and energy will be released, and it will become ADP and this little phosphate group. And then what will happen is energy can be brought back in, and this ADP with the phosphate group can be combined together to become ATP again. So they work in like a circular nature there. On the next slide. Yeah, these are my lovely handwritten notes that I took a screenshot of. To be honest, after we learned this, I really don't remember seeing it a lot throughout the year. But again, a lot of it's like a chem concept or a physics concept, that like energy is neither created nor destroyed. And also when energy is being converted, there's always going to be some energy that is lost. So it's never going to be 100% efficient. So whether that's being like released as heat or something like that, it's never going to be 100%. This gave me flashbacks to chem when we did it, especially the little diagram on the right-hand side. You just need to remember that if instead of delta H, they just happen to have delta G here, which stands for free energy. But you just need to remember that if the value of delta G is negative, I don't know if I've written my notes wrong here. I should probably check that because it is different. Oh, no, well, let's look at the left-hand side. So we have delta G equals your total amount of energy, take away the temperature times S, your entropy. So I'm going to check these notes because it's actually the opposite of chem now that I think about it, because when we have like delta H in chemistry, that's like an exergonic reaction. So does any of the other year twos want to chip in here? Does anyone know? Because to be honest, after we learned this, I've never seen it again. Have any of you guys seen it again? Okay, so moral of the story, it's just not really that important, is it? Because to be honest, like they don't really do a lot of like mathematical chem work in med. Most of it is more like the pathways, but you don't really look at, it's not like in chemistry when you have like your thermochemical equations and you need to know like your delta H and stuff like that. The most important thing to know is to just start getting in the habit of realizing what reactions are going to release energy and what reactions are going to require energy, which they point out when you learn the reactions anyway. On the next slide, it's basically bio/chem. You look at it a lot, and that's just enzymes. And this is more important, I would say. And what you'll realize is when you get into like doing a lot of biochem, there will be a lot of names of enzymes, and they'll always tell you whether like a name of an enzyme is important to remember or whether it's not. And if they say it's important to remember, genuinely remember it, because I remember that they do ask questions on the exams where they can literally just ask you like what enzyme is required for this reaction. What's important to remember is that enzymes speed up biochemical reactions by lowering activation energy. And another important thing is that you have your enzyme and your substrate, and it's important to remember that the substrate concentration affects the rate that a reaction can occur. For example, you could have like an infinite amount of enzymes, but there's going to be like a rate that it reaches and it cannot go above that because all of the substrate like will be filled. So they'll all be taken up and being used. So it doesn't matter if you have even more enzymes because they won't be able to bind with the substrate. And then the last thing about enzymes, which again, you would have probably seen before, is the different types of like enzyme inhibition and things like that. So if we start with part A, that's just showing how our enzymes normally bind to our substrate. Of course, the key word is like the active site. So the substrate will bind directly into the enzyme's active site. Then we have competitive inhibitors, and that is when the inhibitor will bind into the active site of the enzyme, which stops the substrate from being able to bind. And then non-competitive is when it binds to a different part of the enzyme. However, that results in a conformational shape change of the active site, which means the substrate can no longer bind in that area. Most of this shouldn't be too new, but I would say it's like semi-important. I know when we did biochem, they like would ask questions on this in like the little quiz, but I don't think when it came to exams, they really asked questions on it because it's kind of like rote learning. Like once you've memorized what it is, there's not much analysis that they like, you can't really extend your knowledge, if that makes sense. Oh yeah, that's a very good point. It was relevant in pharmacology, like when you're looking at how drugs work and what drugs can be taken at the same time, things like that. Cool.
Okay, so this part, fuels or metabolism, is my section. I'm going to start with a big disclaimer that you're in week 2. I don't think I understood this for weeks and weeks, and like it's confusing. It's like a lot. but you have so many lectures on it, you have so much time, so please don't stress. It's a lot, but you'll understand it. And like, you got it, it's hard. I remember for me, I like looked at it really like there's like individual details, but once you start seeing it as a big picture, that's when it actually, you're like, oh, I actually get this. And then it's fine, because then you can think it through and work it out, and it's less rote learning. But yeah, we can go on to the next slide. This is just a shout out to PSPablo, which were a PSP in the year above us. They're awesome. They made this concept video. Highly recommend you watch it. Not yet, because you haven't learned it all yet, but like later. And once you have learned all this stuff, also just draw this diagram out. Obviously, like your own kind of interpretation of it, and just draw it a million times until it all makes sense. And you don't need to look at any notes to draw it, because you've just memorized it all. But yeah, please don't stress yet, because you don't need to know all this yet. Yeah. And go back one slide. Okay, so what even is metabolism? Essentially, Nirma, what allege, is just trying to teach you what happens with energy in our body. How do we make energy? What happens when we eat food and that contains energy? What happens to that energy? And what happens when we don't eat food but we still need energy? Essentially, it can be broken down into what happens to lipids, carbs, and proteins when we're starving or need energy from our body, or when we're fed and we have extra energy, or we have energy to spare. Yeah, so starving is just the opposite of fed. So anything that happens when you're fed or you've got extra energy, how it gets stored, it then goes the reverse way to become usable. So yeah, and then the other thing, I've gone through a lot, but you have a big lecture called Integrated Metabolism in week 4. That's when everything gets put together. Hopefully by then it will make sense to you. But yeah, let's keep going. Got lots of like little notes. Yeah. So we'll start with glucose metabolism, which is the center of everything, in my opinion. So glucose originally either comes from eating like starch or carbohydrates, like sugary, also like sugars, whatever, via hydrolysis. Glucose is a sugar. Or from the breakdown or mobilization, which is the main word that you'll use, but yeah, breakdown of stored glycogen. So it goes like opposite ways. Like glucose can become glycogen, and then glycogen can go back to being glucose. Yeah, next slide. So the first step when you've got your glucose is glycolysis, which all you really need to know is 1 glycolysis becomes 2 pyruvate, and we'll also need to memorize also 2 ATP and 2 NADH. This can happen in aerobic or anaerobic conditions, so with or without oxygen. What happens with the ATP and NADH comes later, but they're just like useful byproducts at this stage. Hold on, okay. But the important part is that 1 glucose goes to 2 pyruvate, and we're going to follow the path of that pyruvate from there. Just as like just so you know, it is like a 10-step process. It's not just glucose to pyruvate, but the way that you should think about it is glucose to a million other things, and eventually ends up as 2 pyruvate for everyone. Yeah. So now what happens with that pyruvate that we've just made? Also, pyruvate's a big word. I don't honestly really know what it means. It's just a thing that comes out of the glucose. Yeah, it's the broken down glucose into 2. So this is a catabolic reaction. Sorry, this is when we want to, this happens during catabolism where we want to use the energy rather than store it, okay? So we break the pyruvate down. It's either aerobic conditions, which is like normally, like at rest, I guess. We'll break 1 pyruvate into 1 acetyl-CoA, which will become your best friend, but initially will be your biggest enemy. 1 NADH, again, forget about the NADH for now, and 1 CO2, which, to be honest, forget about in general, just know that it makes that. The enzyme for this is pyruvate dehydrogenase. That's an enzyme you do have to remember. And this occurs in the mitochondria. And then if there's no air, so if it's in anaerobic conditions, so that's like when you're exercising, this can happen, or like at the start of exercise, you'll have a whole big thing on this, so don't worry too much about when it's anaerobic or when it's aerobic. Eventually you'll learn. But the pyruvate becomes lactate, or another word for that is lactic acid, And the enzyme used is lactate dehydrogenase, and this produces NAD+, which is also, you'll find is important when you learn alcohol metabolism, but for now, just know that that's what it produces. Also, I feel like I'm just going very fast through everything, so just stop me, you can unmute your mic, you can just write things in the chat, whatever. Tell me if I'm going too fast, tell me if you have any questions. Yeah. So the next thing is the most iconic thing in first sem, but also kind of like a small thing, but just iconic, the citric acid cycle or the Krebs cycle. So firstly, important to note that this only happens in aerobic conditions, and it's where all the acetyl-CoA goes. So when you start learning, we'll go through lipids next, or when you also learn about proteins, essentially everything ends up as making acetyl-CoA, and then it goes into the citric acid cycle to make energy. So the equation is 1 acetyl-CoA, 3 NADH, 1 GTP, and 1 FADH2. So firstly, think of the GTP as an ATP. It's not actually the same, but essentially it is an energy form that we can use straight away. I think it may have a little bit less energy, but you can just pretend it's got the same amount because that's always how I thought of it. And then the NADH and the FADH2 will then enter the electron transport chain, which I'll speak about later, to make ATP. So all that includes this NADH and FADH2, but also the NADH and FADH2 that we made in glycolysis and what happens with the pyruvate. Yeah. So yeah, we can go to the next slide. Okay, so this slide is about the mobilization of stored glycogen, which is essentially so this is now we're in the fasting state. We want energy, but we haven't just eaten. We don't just have glucose available, so we have to make it. So how we got the glycogen in the first place. Firstly, you'll go through it more. You have a bit, but you'll go through it more. But essentially, when you have spare, when you have spare glucose, it can then be made into glycogen and you have extra energy. And now it's okay, now we need it because we're in the fasting state. So let's transport it back, or whatever, make it back into glucose. So firstly, the equation at the top is really important. So it goes from glycogen to glu-1 phosphate or G1P into G6P. Okay. And then depending on where in the body this glycogen is stored, it depends on how we use the G6P. So if you have glycogen in your muscles, it can go straight into glycolysis and essentially start the whole thing that I've just been talking about again. But it can only be used in that muscle, or for that muscle contraction. And then if it's in the liver, the G6P can't go straight into glycolysis. Don't worry about why, just remember that that's a fact. And essentially it therefore needs to be hydrolyzed back into glucose and then enter glycolysis and again. Yeah. So firstly, is there any questions about glucose metabolism before we move into glucose? I'll just move into lipids and just interrupt me all right with that. So lipid metabolism is, firstly, you have a lecture just devoted to lipid metabolism where you learn other stuff, but also this. So next week we'll give you more info and that lecture will as well. But I'll tell you what you need to know, I think for now. So lipids come from either eating them, what foods have fat, and then or from the breakdown or mobilization of stored lipids, which it's stored as tags, triacylglycerides or acylglycerides, however you want to pronounce it, in adipocytes, which are fat cells. Yeah. So firstly, just lipids versus carbohydrates, just this is mostly what you need to know, is that while lipids take longer to break down, they produce more energy per molecule, and so they're good for long-term energy needs. So if you're doing I don't know, it won't be if you start running, when you first start running, you're not using your lipids yet, but when you keep going for a while, then eventually your lipids start burning. Whereas carbohydrates are like fast release, short-term energy needs, but less energy per molecule. So mobilization of stored fats. So yeah, this is when same as like mobilization of stored glycogen. It's when you're in the fasting state, when you need more energy and you don't have it from your food. So it's triggered by glucagon, which is a hormone, which activates lipase, which is an enzyme, to break down tags into free fatty acids plus the glycerol. So you probably would have done these tags in chem. So you've got like the glycerol, and then you've got like each of the fatty acids like that. Yeah, you can picture it. I don't know. Or maybe I'm just talking garbage. But anyway, this part's not as important, but the glycerol then undergoes something called gluconeogenesis, which if you break down that word, glucose, neogenesis making. So essentially it becomes a new glucose from something that's not glucose. And then that undergoes glycolysis. But that part's not as important as beta oxidation, which you will learn about 700 times. It's really important. The fatty acids become acetyl-CoA, NADH, and FADH2. Yeah. So now we've got all this NADH and all this FADH2, but our body can't use that yet. So we need to make that into ATP. So that's what we'll go through now, which uses the electron transport chain. Disclaimer, this is confusing. Honestly, even if you read your slide, you'll be like, oh, she doesn't even know. So yeah. Essentially, it is a way for all the NADH and FADH. We're almost at the end, by the way. The ETC is a way for all the NADH and FADH2 to be converted into ATP, that I've already said, energy in a form that we can actually use, or our cells can actually use. It occurs in mitochondria. Thank you, Gibby. Mitochondria is definitely the powerhouse of the cell. We love that. Essentially, it creates a proton gradient, buzzword, important words, which is essentially like a pressure differential for the H plus, and proton is the same thing. So it's a pressure differential for H plus to cross across this inner mitochondrial membrane. You need to know that. And the way that it does this is using 4 protein carriers, which reduce NADH into NAD plus. So that happens in complex 1, and FADH2 into FAD, which happens in complex 2, and then pumps across the membrane those protons that it's reduced off. Reduce is like a fancy word. I probably understood it during Chem 3-4, but then by the time it started, I didn't. It's just like literally take off the H pluses using electrons. Like that's what you need to know. You don't need to know it all in detail. Complex 3, honestly, they tell you what it does. Someone can back me up if they actually know what complex 3 does. I'm not sure.
Yeah, I can take that. Yeah, pretty much. Complex 2 isn't able to pump the protons out. It's not actually a pump. So pretty much all complex 3 is a proton pump. So it just covers sort of what complex 2 can't actually do. Whereas complex 1 is also a proton pump, so it doesn't need this extra complex. That's all it is. It's not that important. You can sort of like couple it with 2.
Awesome, that was really helpful. Thanks, Libby. And then complex 4, essentially, also I don't really understand what it does. So again, Libby, if you want to jump in, but it produces H2O. That's like the higher important thing.
Yeah, pretty much all it does is it uses now those H plus ions that we just have spare, and uses oxygen. It gets oxygen, they make water, and that's pretty much why it's called respiration, because respiration is making water. So just yeah.
Yeah, and then ATP synthase, another enzyme that you do need to know, then converts the energy from the H plus, which is moving down the proton gradient. So essentially, because there's this proton gradient, the way I think of it is then there's like when it moves down, that like lets energy out because it goes back into equilibrium. So like it's taking energy for them to stay not in the proton gradient. So then when it comes back, then it like releases and it's no longer using this energy, and the energy comes out because, as we know, energy can neither be made nor destroyed, or whatever that fun buzz thing is from school. I feel like maybe we go over it in unison. Anyway, and that energy is used, as Isa said, to make ATP from ADP, and the little thing they write is PI, it means phosphate. Honestly, I probably said that for a while, being like, what is PI? It's just another phosphate. If you think of it, ATP stands for like something like adenosine triphosphate, yeah? And so triphosphate, 3 phosphates, and then ADP is adenosine diphosphate, so 2 phosphates plus this extra phosphate, and joining it up, when you make that bond, that takes energy to make that bond, but then that energy is there for when you break the bond to be released and used by cells or by your body. Yeah. Is there another slide? Oh, yeah. Okay, I'm pretty sure this is the last slide. Or it's not. Essentially, electron transport chain maths. Don't we all love maths? Sounds sarcastic, but I seriously love maths. So 4 H pluses pumped across makes one, all of this you need to remember, makes 1 ATP. 1 NADH gives 10 H pluses or protons, so that equals 2.5 ATPs per NADH. 1 FADH2 makes 6 protons or H pluses, which converts to 1.5 ATP. Because of the way we get from the ATP, from the H pluses to the ATP, is the 4 H plus equals 1 ATP. Okay, so see if you can follow the whole metabolism pathway to work out how many ATP are produced for 1 glucose and for 1 16 carbon chain fatty acid. The reason 16 is because that's the tags are stored with 16 carbon chain fatty acids, which having your mind blank, or whatever, that fatty acid. A couple of other notes. Sorry, it makes me feel like a teacher reading this out. But yeah, it's actually helpful to just like go through it all, and like that means you know all the equations, you understand where everything's going, you know, so it's helpful. Amir, I don't know. Is there anyone else who knows?
Oh, for the question, yeah. So pretty much, it's pretty much not 1 NADH is making 10. It's more like, I think there are 10 NADHs made in the process, and so they're giving 10. And then whereas with FADH2, you sort of get 3 of them being made, and each of them donate 2, so you get 6 total from them, sort of thing.
Yeah, that makes a lot more sense. Thank you.
Okay, yeah, you're right. Sorry, that was me being an idiot.
That's all good.
Yeah. So it's from 1 glucose. Yeah. So the process is from 1 glucose to this. It's like it makes 10 NADH, yeah, and 3 FADH. But yeah, it is helpful. Then you won't be dumb like me, and you'll see that it's 1 that you make 10 NADHs. Anyway, yeah, good luck with it all. Please don't stress about metabolism yet. It will all come together and make sense.
Yeah, so that's the end of our slides and so our session. But like as Eliza said, pretty much metabolism is a big unit, and you're gonna have like 4 weeks on it. So it's like, don't even stress right now. It's good to have a general overview of what's going on and understanding it. But like the amount of tutes and the amount of lectures you'll have on the stuff just will solidify all the concepts. And yeah, as Eliza said, you sort of look at it as a big map and how things interact with each other. But yeah, other than that, any questions, any big thoughts? I'll stop the recording, but yeah, go ahead.